We'll continue on the theme now with a pretty exciting, I think, transition. So Richard was talking about little, oscillators and these sort of simple devices. I mean, Lulu, I'm still amazed. The first talk that I saw Lulu give at Caltech, she described building full-blown circuits out of DNA, and since then she's been able to do neural networks and robotics and these sorts of things out of these DNA computers. And in some sense, you can go to the next level from cutting and pasting in Word to actually having compilers where you write code, and instead of providing assembly code, providing the systems for building these sorts of DNA constructions, computing with DNA directly. So Lulu.
Okay, thank you, Adam. So first, I apologize. I'm the only one who's using this podium because I actually have 70 slides, and if I don't see my notes, I tend to go over time. So that's what I'm trying to do to get everything under control. Okay, so my name is Lulu Qian, and I'm Assistant Professor of Bioengineering here at Caltech, and I'm also affiliated with computer science. So today, I will tell you a story about a future written by molecular programmers. Here's a simplistic view of how do humans actually change the world. We understand things, and we build things. On one hand, we build things to better understand things, and on the other hand, we understand things to better build things. So what makes it easy to build things? Well, my answer is programs. Hardware is controlled by software. Software is programmable, and everyone can write a program. So in some point of view, life itself is a program, one that is written by nature and carried out by molecules. So one genome with a specific set of sequences would produce a bacterium, another would produce a plant or an insect or a mammal. So as a programmer knows, the programming space can be infinite. There are over 1,000 new iPhone apps per day. And we certainly do not have the same number of new molecular apps per day. So it might be ambitious to ask this question. Can humans access the entire programming space of molecular systems? Well, I would argue that it's not impossible if we can actually understand not just what molecules do, but what molecules are capable of doing. So you've seen these pictures many times already today. This is an example of saying electronic machines allow us to send the machines to places that are perhaps too far for humans to go to. And if we truly master the ways of programming molecular machines, then we might be able to build molecular machines and send them to places that are too small for humans to go to. One important concept that enabled the success of computer engineering is hierarchical approach. So from very simple electronic components, such as a transistor, to complex systems that can be designed with the help of programming languages. So if we want to build ever more complex molecular systems, we should also take this concept to develop simple and standard molecular building blocks, and then to understand the behaviors of these molecules as systems, and to design these systems also with the help of molecular programming languages. So I'm not the only one who's excited about this dream. In fact, almost 10 years ago, the molecular programming project, one was the first NSF expedition in computing, was started here at Caltech. Eric Winfrey was the lead PI, and there were 4 other faculty members at Caltech, including Richard Murray, who we just heard from. And 5 years later, the team grew to 11 faculty members, myself included, and was awarded a second expedition. So today, I will try to give you a flavor of where we are in this grand dream. Let's start by looking at the simplest form of life, a bacterium, and ask the question, what types of functional components should a molecular program include? Well, a bacterium has structures, such as the cytoskeleton that helps define the shape of the cell. It also has motors, such as flagella, that enable the cells to swim from one place to another. And then it has circuits, such as genetic regulatory network that produce proteins to do the right thing at the right time. Accordingly, in the field of molecular programming, we use DNA as an engineering material to develop programming languages, algorithms, and architectures to create self-assembled structures, molecular motors, and biochemical circuits. Why DNA? Well, first, DNA is an information-bearing polymer. Second, DNA molecule alone, without proteins, can actually carry out a surprisingly wide range of functions. And these functions are highly programmable, just based on the DNA sequences. But more importantly, models for rationally designed DNA systems have strong predictive power. And finally, it is actually possible to convert other type of molecular signals to and from DNA signals. Just like light, heat, and other types of signals can be converted to and from electronic signals. So the first example I will give you is programming structures with DNA. Well, you know that the natural DNA structure is a double helix in which the sequences of one strand is complementary to another strand. But in 1980s, Nat Seaman was the first to create an unnatural DNA structure. So he actually designed 3 different DNA strands, each of which half of the sequence is actually complementary to the half of its neighbor sequence. So these 3 strands come together to stick to form a 3-arm junction. A much more complex unnatural DNA structure is called DNA origami, invented by Paul Rotman, also here at Caltech. And this technique can fold a circular, single-stranded viral DNA of over 7,000 nucleotides into sophisticated shapes and patterns with the help of hundreds of short strands that bring together different locations on the viral DNA. But the size of these shapes and patterns are relatively small, as Richard told you. It's actually, if you look at the scale bar, it's about 1/10 of the size of a bacteria. So on the other hand, arrays of DNA origamis have been used to create patterns on a larger scale. But because the building blocks is just repeated everywhere, so the complexity of these larger structures are actually quite limited. So how do we actually program complex and larger structures, but still with the nanometer precision? So this was one of the questions that I was thinking about when I started my lab here at Caltech about 3 years ago. And Greg Tikamirov, who's here in the audience, was the first postdoc who joined my lab. And he was a chemist, interested in synthesizing unnatural molecules as complex as what nature can do. And then Philip Peterson joined my lab as a graduate student. And he was an electrical engineer who became an MD PhD student, quite unusual, right? And on top of that, he was an awesome programmer. And before I knew it, the two of them just naturally teamed up and started to making DNA nanostructures in the lab. So this is just one of the beautiful pieces of work they have done. And they used stochastic algorithm to create complex patterns on the surface of self-assembled DNA nanostructures. And these structures are now similar to the size of a bacterium. So we took inspiration from a classic Touché tiling. So this is a very simple tile that has 2 quarter circles, each connecting the adjacent 2 sides of a square. And if you rotate the tile by 90 degrees, there are 2 possible orientations of the pattern. And then if you allow a random orientation of the tile at each location in an array, it generates emergent patterns that can be actually non-trivial to predict just based on the local pattern on the tile. And importantly, every array with a different random choice at each location will generate a completely different global pattern. So generally speaking, a Touché tile has a rotationally asymmetric pattern that is designed to continue into neighboring tiles to form larger patterns. Okay, if you never heard about Touché tiling, but these board games may look a little more familiar, they're actually inspired by the same kind of principles. Okay, so we first folded DNA into a square tile, as you can see from the atomic force microscope image. We then designed the interactions between these DNA tiles to create arrays, each consisting of hundreds to thousands of individual tiles. And with double-stranded extensions on the surface of the tile in the pattern of 2 arcs, and with a random choice of the 2 possible orientations, we created complex and diverse loop structures on arrays of DNA tiles. So here are just the 2 examples of the structure. We colored the original atomic force microscope image on the left to show continuous paths of the loops on the right. And then we further developed a programming language that includes a set of rules for designing the patterns on a tile the probabilities of different choices, and how specific pattern orientations can be adopted at different locations in an array. And then we chose 3 examples, loops, maze, and tree structures, to demonstrate that many properties of these structures can actually be controlled by simple local rules. The design process starts with a high-level description of the tiles and arrays, which can then be translated to abstract array diagrams and numerical simulations. Then to DNA origami tile design, including how the tiles interact with each other on their edges. And finally, to DNA sequences. With these DNA sequences, it is straightforward to just order the DNA strands, as Richard told you, and copy and paste, and pay money, and then mix them in a test tube. And then you get to observe these beautiful images of the designed structure. OK, next example, circuits. Well, how do we program circuits with DNA? So we use a very simple mechanism called DNA strand displacement. An input signal can bind to a gate molecule, and then the 2 strands will compete for the same complementary strand, and then an output signal will be released, but only when the input is present. So using this mechanism, about 5 years ago, I made a DNA-based neural network that can recognize 4-bit patterns that are single-stranded DNA signals. And obviously, that was not smart enough for many of the tasks that you might want to program a molecular machine to do. So when Kevin Cherry joined my lab as a graduate student, he said, let's do at least 100-bit patterns. Well, that's not easy. Because to go from 4-bit to 100-bit pattern recognition, one cannot just use what was developed before. New building blocks and new circuit architectures are required for DNA-based neural network to scale up. But Kevin was successful. He actually scaled up the molecular pattern recognition to 100 bits. How did he do it? Well, we used a simple competitive neural network model called winner-take-all. So here's a function. You have a number of inputs that are real values, and a binary output is 1 if and only if the corresponding input is the largest, which means the output indicates who the winner is. And then you add a layer that computes a weighted sum of a number of inputs before the winner-take-all layer. That allows pattern recognition. So for example, let's consider an input pattern that consists of 9 binary numbers. And if we put the numbers in a 3x3 grid and use black to indicate 1 and white to indicate 0, so you get the input looks like a shape L. And then next, we can simply use the patterns that we like the circuit to recognize as the weights. So in this example, we want the circuit to be able to distinguish a letter L from a letter T. And then these are the weights. And when the input is L, the weighted sum are computed as 5 and 2. And because 5 is larger than 2, so the winner-take-all layer should compute that y1 is 1 and y2 is 0, which means the input is recognized as a L. And if the input is corrupted, in this case meaning that it doesn't look like a perfect L, but the circuit can still recognize it as L because the weighted sum for comparing to L is still larger than the weighted sum for comparing to T. And because of how simple the circuit function is, there actually exists a set of also very simple chemical reactions that can implement the winner-take-all circuits. So I'm not going to explain all these reactions for the interest of time, but I will tell you about one reaction, the pairwise annihilation that allows molecular species, in our case DNA strands, to compete with each other until there is only one winner left. So in Kevin's circuits, there's a mostly double-stranded DNA molecule that we call annihilator, and it can bind to a single-stranded DNA signal because the DNA sequences of the orange domain on the signal molecule is designed to be identical to the orange domain on the annihilator. So they compete with each other for the complementary strand. And if signal Si is present, this process, if only signal Si is present, then this process is actually reversible. It doesn't go anywhere. But if both signal Si and Sj are present, then Sj can also bind to the annihilator, now from the other side, opposite side of this molecule. And then when both signal branch migrate to meet in the middle, and the annihilator will actually split apart, and both signals will be turned into waste product that cannot interact with other molecules. So I'll skip a bunch of details and just tell you the most exciting experiments that Kevin did recently. So this is a circuit for recognizing 100-bit patterns. And to make the example patterns visually interesting, Kevin chose a bunch of handwritten digits from a well-known database that is often used for testing machine learning algorithms. But keeping in mind that each of this pattern actually corresponds to a set of DNA strands. And the circuit is designed to recognize if a given set of clue is more similar to one molecular event or another, but only visually illustrated as 6 and 7. So as I mentioned before, a simple way of remembering patterns is with the winner-take-all circuits is just to use the target patterns as weights. So here, we take the average of 100 6s and 100 7s as a weights, because everyone's handwriting is different. And then each bit in the weights correspond to 2 DNA molecules that perform the weight multiplication function. And the concentration of these molecules corresponding to the value of the weights. So in this database, there are a total of over 10,000 example handwritten digits 6 and 7. If we look at where the example pattern are in the weighted sum space, it tells us how easy or hard the winner-take-all computation should be. Further away from the diagonal line means it's easier, and closer to the diagonal line means it's harder. So 1% of these patterns are actually on the wrong side of the line. So it's impossible for our circuit to actually recognize these patterns correctly. And 6% of these patterns are fairly close to the diagonal line, which we expect to be experimentally difficult. And then 94% of these patterns are far enough from the diagonal line, which we expect our circuit to be able to recognize correctly. So Kevin chose 50 examples from these patterns to perform his experiments. And the perfect patterns, which are just the same as the weights, but in binary, were computed correctly. So if you look at the green trajectory going high, that indicates 6, and the yellow trajectory going high indicates 7. And more importantly, corrupted patterns were also recognized, up to more than 28 bits of corruption. That's the beauty of the circuit. OK, let's finally move on to the example of motors. So when Anu Subagir joined my lab as a graduate student, she told me that she was interested in DNA circuits on structures because she believed that the combination would actually do more than circuits or structures alone. So I said, oh, maybe we can work on DNA robots together. So I did. Anu built a DNA robot that can sort multiple types of molecular cargos that are initially randomly distributed into piles of distinct molecules. We used a very simple algorithm. So the robot performs a random walk. If it bumps into a cargo, it will pick it up and continue to perform random walking. And if it bumps into a goal location while carrying a cargo, and if the cargo type and the goal type match each other, and then the cargo will get dropped off at the goal location. So this algorithm does not require the robot itself to be very smart, and because the recognition is encoded between the cargo and the goal location. So here's what our robot looks like. It is just a single-stranded DNA molecule that has 2 foot domains and a leg domain. And again, using the strand displacement mechanism, it can move from one location on the track to another. And the track is actually built on a DNA nanostructure surface. So using a similar mechanism, we add an arm domain and a hand domain to the robot for picking up and dropping off the cargoes. The recognition between each type of cargo and goal is simply encoded in the complementarity of DNA sequences. So here's the experimental setup that Anu designed. The 2 types of cargoes are initially mixed together near one edge of the DNA nanostructure, and then the 2 separate goal locations are near the other edge of the DNA nanostructure. And the bright spots that you see in the atomic force microscope image indicate cargoes at the goal location. So we used distinct fluorophore molecules as cargoes and labeled the goal location with quenchers. That means when the cargoes are dropped off, the corresponding fluorescence signal will decrease. And from these 2 sets of data, you can see that cargo 1 was successfully dropped off at goal 1, while cargo 2 successfully dropped off at goal 2. OK, so this is, of course, not reality yet. But imagine that. Perhaps one day, DNA robots can actually be used to break down trash into individual molecules and then sort them into distinct piles for recycling. OK, so I've talked about 3 examples of how programming languages, algorithms, and circuit architectures can be developed to program molecular systems using DNA. I hope these examples have given you a little bit better idea of how programming molecular systems could change the world in the future. We used to think that there are many differences between living things and non-living things. For example, living things can move, learn, and grow, and reproduce, and evolve, etc. But with computer engineering, non-living things now can also move and learn, and in some cases, even better than living things. So perhaps eventually, when we actually master the ways to program molecular systems, the differences between living and non-living things will become even smaller, and all things will be programmable. If you're a fan of Harry Potter, then you must have heard about Hermione's bag. She travels with a very small bag, and then she pulls out clothes and many other large items from the small bag. So perhaps this is not impossible in the future. We might be able to program a seed that grows into a chair with a specific light shining on it, and then the chair will shrink back into a seed with another different type of light, so you can travel as light as Hermione. I know, it still sounds crazy today. That's why I feel very lucky to have a group of excellent people who are excited to work with me on these crazy ideas that maybe one day can change the world. Thank you.